So just to start, I'd like to cite Crick and Jones back in 1993, who said the following at that time. What is known about the neuroanatomy of the human brain? Do we have a cortical map? And what does the human connectional map look like? The shameful answer is that we do not have such detailed maps. It is intolerable that we do not have this information for the human brain. Without it, it is little hope of understanding how our brain works, except in the crudest way. Clearly, what is needed is a modern human brain anatomy, is the introduction of some radically new technique. What a better introduction to this brain competition, I would think. So, what I'm going to do in these 10 next minutes is to give a very brief overview of what has happened basically since this citation in the mid-90s and now. And this will take us through diffusion imaging, tractography, and MR connectomics. So back in the mid-90s, there were these early experiments on DTI, and the people noticed at that time that diffusion is actually anisotropic in the white matter of the human brain. From there, we've gone quite a bit forward, and by better understanding the diffusion MRI physics, but also improving our hardware tools and pulse sequences. And the main result of that was that we got new imaging paradigms going mainly along with higher angular resolution, high B values. Several imaging techniques came up, Q-ball, persistent angular structures, spherical deconvolution, diffusion spectrum imaging, and so on. And these new techniques allowed us to leave the simple diffusion tensor model and be able to characterize more precisely how fiber orientations are distributed inside a single voxel. This is extremely important since, given the current voxel size, there are always several populations of fibers with different orientation inside a single voxel. Nevertheless, in my opinion, there is still a significant room for hardware improvement and MR technology. I believe that stronger gradients, faster imaging techniques, and coil technology will, in the next 5 years or so, significantly further improve our imaging ability of fiber tracts. Nevertheless, MR tractography is intrinsically limited in several ways. And the fundamental biophysics of diffusion tell us that there is an intrinsic angular separation power in diffusion that limits us in our ability to separate different fiber population inside the voxels. There are important sample issues that are difficult to control for. For example, when you're doing in vivo imaging, brain motion, brain pulsation, and perfusion artifacts are very present. And if you do ex vivo studies, tissue degradation is going to be a very important component in these limitations. Furthermore, however, whatever your MR technology is going to be, you're always going to be limited by voxel resolution, angular resolution, SNR, and acquisition artifacts. And aware of all these limitations, this led an open door for multiple image processing methods for tractography. And this is what I call the tractography algorithm jungle. There are tons of different clever solutions to tackle that inverse and difficult problems, depending on what playground for tractography is used, DTI, multi-shell or single-shell techniques, different approaches for tractography. The classic ones are deterministic line propagation techniques, but other people came up with probabilistic tractography, energy minimization techniques, coming from the differential geometric framework or the statistical mechanics. And I'm sure I forgot many other possibilities in this thing here. So when we consider this splay crossing problem in the brain, it is, I'm going just to give you here an example how different results are going to look like depending on the imaging technology you're going to use and the tractography method. So on your left side, you have got a reconstruction of the centrum semiolale with DTI and standard streamline tractography, and we easily identify here the U-shaped corpus callosum. Measuring the same region of the brain with a high angular resolution technique, which is in this case diffusion spectrum imaging, and reconstructing the fiber trajectories with a streamlined fiber tracking, you end up with a solution like that, where you've got where actually you resolve quite nicely the crossing in this area between the corpus callosum and the corticospinal tract. But it's not obvious that this is the right solution, or this is the right solution, or maybe this might be the right solution. Here we've got an example where energy minimization technique has been applied to the same region, and we see that there is complex intermixing of crossing and splaying fibers, and it's not very clear what's going to be the right answer here. Obviously, better SNR and better resolution is always good. Here, a striking example of the pons of the human brain acquired on one side ex vivo here, and compared to the in vivo equivalent here. And so at the end, what we really all strive to is to have just exquisite images of the fiber anatomy in the brain. But what does fiber exquisite reconstruction mean? And this brings me to the question of validation. Yes, this damn word. So, the big difference between the brain and the rest of the body is that actually we don't know what the anatomy is like. We have no gold standard. And what we are left is several partial tools to try to get a feeling of what we're going to examine. So is the eye of the trained neuroanatomist going to be enough to say, well, this is a good measure, this is a good reconstruction? Or do we need to do acquisitions in the macaque and compare it to some database of fiber tracing studies like COCOMAC? Or do we want to compare structural connectivity with functional connectivity in order to get a sense if we are doing a good job? Well, my opinion is that since we have no gold standard, there is a necessity to explore a combination of this, or to validate your results with a combination of these different approaches in order to get a feeling of what you're doing. And obviously, I forgot this slide, where obviously standard functional imaging is also going to be a very important component in order to validate your tractography design.